guys, Rob from Royal Balls. Today we're going to talk about genetics. It's a apparently very complex subject. Many people have attempted videos. I'm going to try and simplify it. I'm not a geneticist, but I am a scientist, and I do like to try to simplify things to make it easy to explain. So we're going to do a three-part series on genetics. The first part is going to cover single gene combinations. The second part is going to cover multi-gene combinations, and the third part is going to cover reverse engineering. What snake do we need to make? In this case, I'm going to have a reveal of a snake that I've recently introduced into my collection. So I'm going to reverse engineer what genes are needed to create that snake, and then what that snake can produce in terms of its offspring. So three parts, just to keep it nice and short. So let's take a look. Okay, let me kick this off by showing you my genetic calculator. I think you'll agree that's fairly simple. There are only 4 possible outcomes, so this should be fairly easy to follow through. Anybody can remember 4 potential outcomes. I know what you're thinking. You're going to say that there are millions of different combinations of all the various genes that are involved in ball pythons. That is true, but they all boil down to 4 combinations. You can use this as a genetic calculator far more easily than you can compute millions and millions of potential outcomes. Let me show you how it works. I think everybody remembers from Jurassic Park, the original Jurassic Park, where Mr. Hammond recreated dinosaurs, and he had a little video that he used to intro for his guests, a little cartoon character called Mr. DNA, and this is Mr. DNA. This DNA strand here contains all the material necessary to recreate, in his case, a dinosaur, in our case, a ball python. He extracted the DNA, which was incomplete in his case, and he filled in the genetic blanks using DNA from a frog to fill in the blanks, and he recreated dinosaurs. That process is known as cloning. What you're actually producing there is an exact copy of the DNA that's present in the dinosaur that it came from. In our case, when we're breeding ball pythons, we're going to use the DNA from both parents. So both parents provide exactly half of the material required to build a new snake. If you remember from the cartoon, Mr. DNA was a double helix. It was 2 strands of DNA wrapped around each other. So this genetic material, or DNA, controls everything about the snake. There's a complete copy of everything needed to recreate the snake's digestive system, its head, its eyes, its reproductive system, everything about that snake. Only a very, very small piece of this genetic material controls the colour. So there's a small piece of DNA or code within that genetic material that controls colour. So this is our blank canvas for ball pythons. This is going to be the normal, classic or wild type. There's no material in here that changes that basal colour. Fortunately, nature provides us with a lot of different variations of colour schemes in ball pythons, and they're commonly called morphs. Morph is morphological. All that means is a variation of the structure of the animal, in this case the colour, and the thing that controls the variations in colour of the animal is called a gene. Just as in humans we have genes that control eye colour or hair colour, facial features, so our ball pythons also have genes which control variations in colour and pattern. So just like a computer code that inserts itself into the big computer program, the piece of genetic code that goes in here or attaches itself to that genetic material is called a gene, and there are many, many different types of genes. So each of these genes is like a piece of a jigsaw puzzle. It only fits in one particular slot. on the genetic code. And in this case, this gene is going to slot somewhere into that piece of genetic code that controls the colour and pattern of the ball python. When we breed ball pythons, the male and female take exactly half of their genetic material and pass it on to their offspring. So here we have the female and here we have the male. So the male has split his genetic material in half. There's only half the required information in here to build the new snake. He combines that with half the material from the DNA of the female. So that is now our new snake. Okay, so we have male, we have female, we have two halves, 4 possible combinations. This half of the male to this half of the female, or this half of the female, or this half of the male to this half of the female, or this half of the female. Only 4 possible combinations. So let's go back to my genetic calculator. Those are the 4 possible combinations. What we're going to do today, if I just move these down out of the way, is we're going to concentrate on a single gene, and we're going to use one of the common genes, one of the codominant genes. We're going to use pastel, but it could be enchi, it could be yellow belly, it could be any of the single gene codominant combinations. Now, you've probably heard the term codominant. Incomplete dominant is probably a more accurate term, and I'm going to explain why in a moment. Okay, so we're going to breed a pastel to a normal, and we have 2 snakes that are intertwined like DNA here. So let me just see if I can sort them out. So we have a normal, which is this guy here, the normal pattern, and we have a pastel, which I think you can see is it's a pattern and colour mutation of the normal. So this animal here has 1 gene in it that the normal animal does not. So it's a single copy of a single gene, and we're going to model that using my genetic calculator. So let's get rid of the snakes before they destroy the set. Okay, so let me bring back my genetic calculator and bring back my strands of DNA. So I said the sex doesn't matter because genetics in general are not linked to the sex. So it doesn't matter when I take my single copy of the pastel gene whether I attach it to the male or whether I attach it to the female, which means in our pairings it could be the female or it could be the male that's pastel. It holds true for this calculation regardless. So I'm going to attach the pastel gene to the male, and you'll notice that it's attached to 1/2 of his genetic material, and that's because for every address where a gene can reside in the genetic material there is room for 2 copies. You can have 0 copies, you can have 1 copy, or you can have 2 copies. Same for the female, 1 copy or 2 copies in exactly the same address on her genetic material. But for the purpose of this exercise we're going to have the single gene pastel animal, and it's in the male, but that doesn't really matter. Okay, so we've got, as my genetic calculator suggests, 4 possible outcomes. So here's the male with his half of the genetic material, could be paired to this. So 1 copy of the gene from the male, no copies of the gene from the female. The other possibility for this half of the male's genetic material is to combine with that. So again, 1 copy from the male, no copies from the female. Let's take the other 50% of the genetic material from the male, and I think you'll see here that it doesn't matter which one you combine it with, there are no copies of the genes being transferred. So for these 2 squares, 0, 0, 0, 0. So what have we got? We've got 4 potential outcomes for that combination. This is going to be pastel, one copy of the gene. This is going to be pastel, one copy of the gene. This is going to be a normal, no copies of the gene. This is going to be a normal, no copies of the gene. So out of a 4-egg clutch, or 4/4 of the pie if you like, we're going to see two pastels. It's not a very nice 2, is it? And 2 normals. So there is a 50-50 chance in that pairing of getting a pastel or a normal. In a 4-egg clutch, that means you get 2 pastels, 2 normals. In an 8-egg clutch, that means you get 4 pastels, 4 normals, on average. That is a statistical average. Okay, what we're going to do now is to pair up a pastel to a pastel. The same genetic combinations now in the mother and the father. So let me just wipe off this outcome. We're going to use, notice we're going to use exactly the same 4 outcomes, but for a different set of genetics. We've got 2 copies of the gene involved in this reproductive scenario now, but we're still only going to get 4 outcomes. So let me show you how that works. So here's the female. She has 1 copy of the pastel gene. She is a visible pastel. Here's the male. He has 1 copy of the pastel gene. He's a visible pastel. So when they breed, they each provide 50% of their genetic material. You remember I said that in any combination, for this genetic slot here for pastel, the male can have 0, 1 or 2 copies. So when we split the genetic material, half of the genetic material will carry the copy that he has, and half of it will not. And the same for the female. Right, what are the potential outcomes for this? There are only 4 potential outcomes because we only have 4 pieces of genetic material to recombine. So, here's the male. We combine this half with the pastel gene to this half that doesn't have the pastel gene. So we get a copy from the male. We don't get a copy from the female. We can also combine this half with this half. And now you can start to see that there is actually a difference in the outcome. So for this one, you get a copy of the gene from the male and a copy of the gene from the female. So in this outcome, there are 2 copies. Let's take the other half of the male genetic material. We can combine it with this, which gives us no copies from the male, no copies from the female. Or we can combine this half of the genetic material with this half of the female, and that gives us 1 copy from the female, no copies from the male. And they are the only 4 possible outcomes from this pairing. What do we get? Well, we get 1 square is normal, no genes. We have 2 squares that have 1 copy of the gene. So this is pastel, this is pastel. We have 2 copies in this square. So let me introduce a new concept now. For a codominant or incomplete dominant gene, there is a superform. So we can have super pastel. This square contains 2 copies of the gene. So when you combine the male with the female, that particular offspring will be carrying 2 copies of the pastel gene when it's recombined. So that offspring will be a super pastel, and that's what we mean by super pastel. You will also hear incomplete dominant, and I think that's a more descriptive term for what's going on here, because the pastel gene in its incomplete dominant form, you only need 1 copy, and it does change the visual expression in the offspring. So pastel and pastel are visible in the offspring even though you only have 1 copy. In order to see the full expression of the pastel gene, you need the superform. The superform is the full and complete expression of that gene. So this is the superform. Okay, so let's look at the probabilities in this outcome where we're pairing a pastel with a pastel. You can see, again, the 4 squares, each representing 25%, or 1 in 4 of the eggs from that particular pairing. If we look at this square here, this is the normal. There are no copies of the gene in this square. So 1 in 4 of these babies will be normals. Even though you've paired a pastel with a pastel, you will still get a normal, and there is a 25% chance of a normal. If we come to the pastels, you will see that there are 2 of the squares, or 2 quarters, which is a half. A half of the babies will be carrying 1 copy of the pastel gene, so they will be visual pastels. So you have a 50% chance of getting a pastel baby from this pairing. Come across to this corner, this is now the super pastel. 2 copies of the gene, the full visual expression of pastel, and you will see that that outcome is only in 1 square. So there's only a 25% probability, or 1 in 4 chance of the babies being super pastel. So if you get a 4-egg clutch, statistically you will get 1 normal, you will get 2 pastels, and you will get 1 super pastel. If you have an 8-egg clutch, you will get 2 normals, you will get 4 pastels, and you will get 2 super pastels. So you can see now, even though that we've added a copy of the pastel gene to the pairing, we're now pairing a pastel to a pastel, you can still use the simple 4 outcome permutation, if you like, for what the offspring are going to be. Let me do that again with, let's take a super pastel to a pastel. So let's remove this, and I'll show you again that we're now, let's make it the male, but it doesn't really matter. So the male is now a super pastel. He is carrying 2 copies of the pastel gene. The female is just a pastel. Right, so now we've got 3 copies of the pastel gene in the genetic equation, but still only 4 possible outcomes. So let's work the numbers. Let's rub these out. Okay, so the female splits her genetic material in half, and the male splits his in half, and we're going to recombine them in the offspring. This half of the male's genetic material can combine with this half of the female, so that's 1 gene from the male, no genes from the female. This half can combine with this half, and this half of the genetic material is also carrying the gene. So we get 1 from the male, 1 from the female. We come back to the other half of the genetic material from the male, and because he is a super, he's carrying this gene on this half of his genetic material as well. So this half combines with that half, and that's exactly the same outcome as the line below. 1 gene from the male, none from the female. Combine it with the other half of the female genes. 1 copy from the male, 1 copy from the female. Okay, so still, although we've got 3 copies of the gene now in the genetic equation, still only 4 outcomes. So in this outcome, what do we get? We have a super pastel with 2 copies of the pastel gene to a pastel with 1 copy of the pastel gene. Now you can see that we're not getting any normals. There are no squares here which have none of the genes, so no normals in this clutch. We have 2 squares which have 1 copy of the gene, so 2 squares which are pastel, so that's 50%, that's half of the babies will be pastel. The other 2 squares are carrying 2 copies of the gene, super pastels. So 50% of the babies in this clutch will be super pastels. So you have, out of a 4-egg clutch, 2 babies are pastel, 2 babies are super pastel. Out of an 8-egg clutch, 4 of the babies will be pastel, 4 of the babies will be super pastel. And that is from the pairing of super pastel to pastel. Now that is what we mean in ball python genetics when we talk about the super form. The super form of an incomplete dominant gene means that the snake in question is carrying 2 copies of that incomplete dominant gene, which means he is going to pass on to his offspring, regardless of which half of the genetic material you choose, he is going to pass on 1 copy of the gene to every single one of his offspring. So when you have a super form or a super pastel or a super orange dream, a super enchi, a super yellow belly, all of the offspring will carry that gene. So in this case, all of the offspring from that super pastel pairing will be pastel. Because the female is also carrying 1 copy of the gene, 50% of those babies will be the super pastel because they'll get the copy from the female parent as well. So that is the power of the super form. That's what we mean when we talk about super form in incomplete dominant genes. That's why breeders are so excited about having a super form in their collection, because you are guaranteed that gene will be passed on to every single one of the offspring. Let's do the pairing of a super form to a normal, and I'll show you the power of the super form. We have the pairing of a super pastel to a normal, and again, doesn't matter which of the sexes is the super form. For this equation it works out exactly the same. So we split the genetic material 50-50, still only 4 possible outcomes. That's the beauty of this calculation. Every single calculation you do, there are only 4 possible outcomes. It's very simple. Anybody can do it. So let's do the pairing. Here is 50% of the male to there or to there. So that offspring will get a copy from the male, but no copy from the female. The other 50% of the genetic material from this guy is also carrying the gene. Paired to this or paired to that is exactly the same outcome. A copy from one parent, no copy from the other parent. So there's your 4 outcomes. Every single one of those offspring from this super form, even though you bred it to a normal that is not carrying the gene, every single one of those babies is going to be pastel. That is the power of the super form. Okay guys, so that's the first part of my genetic calculator for people like me who don't like things too complicated. Those are the single gene combinations. Part 2, we're going to focus on multi-gene combinations, and we're still going to use the simple 4 square method. In actual fact, most of the genetics has been done. All that remains is some probabilities, really. So that'll be part 2. Anybody can use Morph Market, the genetic calculator on Morph Market, or the genetic calculator on Ball Python World, and that will give you a very quick and easy way of sorting out what your pairings will produce. But I get frequently asked, why? Why does that snake produce those offspring and not another kind of offspring? Why does that genetic combination produce this? Why does it produce that? What does heterozygous mean? What are hets? What do they do for me? So we'll explain all of that in the second part of the genetics video. I hope you enjoyed the first part. I hope it was simple enough that everybody could understand it. I do encourage you to ask questions, make comments, and if you do have a particular genetic problem that you think would fool my genetic calculator, send it in. I'll work out the pairings for you, and I'll work out the odds for you using my very simple genetic calculator, and we'll see if it works. So thanks for watching. Don't forget to like and subscribe, and we'll see you next time.